Yes, thank you for all showing up a little earlier today. I think John wants to introduce himself because he's really good at it, so go ahead.
Yeah, okay. Hello, everyone. I'm John. Do you mind closing the, shutting the lights over there so that we get nice cinema darkness? There we go. Now I feel at home, nice and cozy. Okay, so can everyone hear me in the back? Yeah. Okay, good. So today I'll talk about detectability of cerebellar activity with MEG and EEG. My name is John Samuelsson. Maybe some of you knew that already. I'm in HST. I work with Matti and Bruce. So the cerebellum means the little brain, but it really is central in many ways. For instance, the majority of the brain's neurons are actually found in the cerebellum. It is one of the most widely interconnected structures in the human brain, connecting with a broad range of cortical regions, not only the motor cortex, but also areas engaged in higher order functions, such as the prefrontal cortex and parietal cortex. Although the cerebellum has traditionally been seen as a brain region mainly engaged in lower motor functions, then especially motor coordination, research over the last 2 to 3 decades has shown that the cerebellum is in fact endowed with a much richer range of functions than previously thought, including emotional and cognitive processing. So, although the cerebellum has been traditionally shamefully overlooked both in neuroscience and clinical research, there is mounting evidence for cerebellar involvement in a wide range of neurological diseases, including some of the most prevalent disorders such as Parkinson's and essential tremor. Parkinson's affects over 1% of all people over 60, and essential tremor affects about 4% of all people over 40, which makes the cerebellum pertinent from a clinical standpoint. Although MEG and EEG have been used to extensively study the neurophysiology of the cerebral cortex, very few studies have been done on cerebellar function, and there is therefore a lot of untapped potential in this field. But that hinges on the critical question of whether it is possible to detect and record cerebellar activity with MEG and EEG. And as far as we know, no one has done an analytical investigation on this topic. So addressing this gap will be our quest here today. So is it possible to measure cerebellar electrophysiology non-invasively in humans? In order to answer this, there are 3 major concerns that need to be addressed. The first is which cellular electrophysiological mechanism generates a signal that we want to measure, since we need to know the alignment, amplitude, and orientation of these cellular neural currents in order to model them. Secondly, the magnetic field amplitude decreases as inverse over distance squared from the source. So the further away the source is from the sensor, the weaker the signal. The subcortical location of the cerebellum, which you can see in the figure here, there's the cerebellum in case you didn't know it, therefore it needs to be addressed. Thirdly, and perhaps most critically, how bad is the signal cancellation due to the tightly folded cerebellar cortex? If the neural currents are oriented orthogonally to the cerebellar cortex, as they are in the cerebral cortex, we will have low cancellation when there is low curvature of the cortex, such as on the figure to the right here, where the orange arrows represent current dipoles that are almost aligned, and their magnetic fields therefore adds up at a distance, and we have low cancellation. But if there is high curvature, such as here, on the figure here, these 2 current dipoles are of opposite polarity, and their magnetic fields therefore nearly cancel out at a distance where the sensors are. Because the cerebellar cortex is so tightly folded then, as compared to the cerebral cortex, as you can see in the image here, we expect a higher degree of signal cancellation in the cerebellum as compared to the cortex. Quantifying the degree of cancellation is therefore of paramount significance when answering the question of whether it is possible to detect cerebellar activity. So the first thing we need to know is what cellular mechanism generates a signal that we want to measure. Dr. Yoshio Okada here at the Martinos Center and Children's, I think, showed from in vitro experiments that the physiological origin of the MEG signal from the cerebellum comes from dendritic currents through Purkinje cells. Dendritic currents in pyramidal cells are modeled as current dipoles and are well known to generate the MEG signal from the cortex. Because a similar mechanism underlies the signal from the cerebellum, we model neural activity in the same way, namely as current dipoles lined up orthogonally to the cerebellar cortex, like the black arrow shown here. Because the dendritic tree is largely flat and spreads out in 2 dimensions, much like a hand like this, the net current will be perpendicular to the cortex like this. And therefore, this here is a good model of the current that generates the signal. So based on these experimental results, we can model neural activity in the cerebellum the same way as we do in the cerebral hemispheres, and then use established numerical methods and software to simulate the signal. But to do so, we need a geometrical model of the cerebellar cortex so that we can constrain the location and orientation of the current dipoles. Conventional MRI, however, does not have sufficient resolution to resolve the folia, as you can see in the image to the right here, which shows a naive free surface reconstruction based on conventional MR data. So here we have used the high resolution tessellation of the cerebellar cortex, which you can see on the right, handed to us from a collaborating lab led by Martinos at UCSD. This tessellation is based on repetitive ex vivo MRI scans of a post-mortem human cerebellum in a 9.4 Tesla magnet. This model is then fitted to the individual source space of the subject by starting with this naive free surface reconstruction, which is subject specific, and then taking our high resolution model and adapting size, orientation, and location to fit the individual source space of the subject. So we end up with the source space seen here with the 2 cerebral hemispheres and the cerebellum. Based on this source space, we can then compute a forward model, which is used to simulate the signal. This was created using NOEM's MMVT tool. Using this forward model, we have performed Monte Carlo simulations to do patch analysis. In these simulations, a vertex is randomly picked on the cerebellar or cerebral cortex. This vertex then becomes the midpoint of a patch that is expanded continuously around it. You can see an example of this in the figure to the right here, where this yellow area is an example of such a patch. The signal is then simulated by activating all current dipoles in the patch for different sizes. We then compute the norm of the simulated signal, which tells us the cerebellar signal strength. We also add the norms together of the signals generated by each dipole activated individually, which is the absolute signal. Because each dipole is activated individually, this metric is unaffected by signal cancellation. We can then quantify signal cancellation by dividing the net signal by the absolute signal. We call this ratio the conservation factor and will be between 0 and 1. That is C in this equation. So beta here is the absolute signal, C is the conservation factor, and alpha is the net signal. So this conservation factor C will be between 0 and 1. If the net signal is the same as the absolute signal, then there is no signal cancellation, and the conservation factor is 1. If the net signal is 0, but the absolute signal is non-zero, we have complete signal cancellation, and the conservation factor is 0. Moving on to results of these Monte Carlo simulations, we have the absolute signal in the picotesla for MEG in the left column, and microvolts for EEG in the right column, plotted to patch radius. The line is the median value, and the bars represents 1 standard deviation. Do you see online? Oh, did it die? Hello. Yeah. It's working all right. Yeah. Thank you for that. So the CN line is the cerebellar data, and the orange line is the cortical data. We see that both in MEG and EEG, the cerebellar absolute signal is about 30% weaker than the cortical absolute signal across patch sizes. That is, the subcortical location of the cerebellum alone makes the signal about 30% weaker than the cortical signal. However, the absolute signal does not incorporate the cancellation effect, which we noted just a moment ago. So to quantify the cancellation effect, we need the conservation factor. Calculating the conservation factor for different patch sizes, which you can see on the right, we see that the cancellation is indeed worse in the cerebellum than in the cortex. But it is not critical. For patches of radius greater than 5 millimeters, the conservation factor is typically 30% lower in the cerebellum than in the cortex in MEG, and a little bit worse in EEG. This difference is even smaller for patches of radius smaller than 5 millimeters. So the net signal incorporates both of these effects, since the absolute signal, the upper row, times the conservation factor, the middle row, gives the net signal, which is here shown in the lower row. Because the absolute signal is about 30% weaker than in the cerebellum, and the cancellation is about 30% worse, the net cerebellar signal is about 40 to 60% weaker than the cortical signal. These results are quite promising. Studies on other non-cortical brain regions typically find that the subcortical signal is orders of magnitude weaker than the cortical signal, whereas these results here show that the signal strength is on the same order of magnitude, albeit about 50% weaker. But these are all referring to median values, and as you can see in these bar plots, the variation in these results are quite high. So how do these results vary over the cerebellar cortex? This is a sensitivity map showing the gradiometer coverage. Enlarging the cerebellum, we see a clear gradient between the anterior lobe, which has poor coverage, and the superior part of the posterior lobe, which has better coverage. To show this better, we can view a mid-sagittal cross-section of the cerebellar vermis, which is plane 1, along with plane 2, which makes a 45-degree angle in the lateral direction of the midline, showing a cross-section of the right lateral hemisphere. So doing a close-up of these cross-sections, we see that the lateral hemisphere, that is plane 2, has generally better coverage than the vermis. We also notice a big difference in sensitivity between the different folia, which can be more easily assessed by marking them. We can also notice the high variation in sensitivity inside each folia, which happens because of the high curvature of the cerebellar cortex. Generally, though, we see that lobule 6, crus 1, and I need my laser pointer now. I hope this is working. There we go. Yeah. So lobule 6, crus 1, and crus 2 have particularly good coverage. Looking at the full picture and comparing these regions to a topographic map of cerebellar functions found by task fMRI studies, we see that crus 1 and crus 2 are strongly associated with working memory here, which is colored here in blue. But these results are only for gradiometers. What about magnetometers or EEG? So here we see gradiometers on the left and EEG on the right. Magnetometers are a bit more far-seeing than gradiometers, but are quite similar, so we'll leave them out now. Note that MEG and EEG have very complementary sensitivity distributions. While MEG has good coverage of the posterior lobe here, EEG has quite good coverage of the anterior lobe here. What all modalities have in common, however, is that they're all sensitive to activity in lobule 6 and crus 1 around here, as we noted earlier. And this corresponds to working memory processing, so we would therefore recommend working memory task as a default experiment for probing cerebellar electrophysiology. So to conclude, the cerebellum has traditionally been overlooked. A study on cerebellar detectability with MEG and EEG has been lacking. To address this gap, we built a forward model relying on previous experimental results and a high-resolution tessellation of the cerebellar cortex. We found that the cerebellar signal is about 50% weaker than the cortical signal. We also found that both MEG and EEG are optimally sensitive to activity in crus 1, which corresponds roughly to working memory, but otherwise rather complementary sensitivity distributions. So we recommend a combined MEG-EEG experimental setup for probing cerebellar electrophysiology. In the future, we will develop a method for mapping cerebellar source estimates in any subject. We will also investigate the limitations and possibilities of cerebellar inverse modeling, and finally validate these results on experimental data using auditory working memory, since that was a good experiment that has high sensitivity with MEG and EEG. So the co-authors or the collaborators, I guess, or my lab colleagues in this study was Padma, Shiraz, Martinos at UCSD, and Matthew, of course. And I would also like to thank Noah and Bruce, Meinak, and Lawrence for help along the way. And that's about it. You can turn back the lights. And, yeah.